Hello there guys, today we'll be talking about insulin. So insulin produced by pancreas also by the islets of Langerhans. I'll look at the beta cells, but beta cells produce insulin, alpha cells produce glucagon. Now for the beta cells, it's produced the same way with glucagon. So mRNA, then ribosome create the protein, Ruffy arrow modify it, Golgi body modifies it further, then you have insulin and your C-peptide. Then glucose goes to the cell, through different processes produce ATP. ATP inhibits this potassium channel. This inhibition causes sensitivity of this calcium channel, allowing calcium to activate the SNP proteins in the vesicle, allowing for exocytosis of insulin, C-peptide, and amylin. And this C-peptide is important for measuring insulin levels. So usually when I measure insulin levels, I actually measuring C-peptide levels because insulin and C-peptide goes together. And amylin, it's a bit more complex than that. Anyway, insulin goes to the bloodstream and acts on the liver, the muscle, and fat cell. Now, in the liver, it activates this tyrosine kinase receptor, which activates a second messenger called PI3K and AKT. Now, this AKT is basically just protein kinase B. Now, AKT will activate glucose to go through the glycolysis pathway to create pyruvates, and also force glucose to go to glycogen. Now, this glucose comes in through this GLUT2 transporter, which is insulin independent, like with the beta cell. GLUT2, insulin independent. Then for the muscle cell, we have your tyrosine kinase receptor again, which activates PI3K and AKT. Now, GLUT4 is insulin dependent, so it needs insulin to work. So this second messenger here activates this channel, allowing glucose to come inside. Now, when glucose come in, it turns glucose to pyruvate, which then turns to acetyl-CoA and other things. Now, in this place, to use pyruvates, we eventually turn to acetyl-CoA and be used for fatty acid synthesis, which we'll talk about. We're talking about the fatty acid or fat cells. And also, this PI3K and AKT also activates the amino acid channel, causing amino acid to go in and also promote protein synthesis. Now, lastly, we have the fat cell. So, same process again. PI3K and AKT is activated, which causes glucose to go to glycolysis, producing DHAP, that is dihydroxyacetophosphate, and D3AP. D3AP will get to acetyl-CoA and be used to form fatty acids, while DHAP will be used to form glycerol. And glycerol plus fatty acid, you get your dry side glycerol, and you now have fats. And that's the end.